Good afternoon, everyone. Thank you for joining us. It's a pleasure to have you all. We have a very special guest today, Dr. Amir Ibrahim, a FAFS alumnus and Distinguished Alumni Award candidate also. So just to give you a brief biography of Dr. Ibrahim, he's the Regents Professor at Texas A&M University and wheat and oat breeder geneticist. He teaches and conducts research on wheat and oats. He released and co-released 18 wheat and 3 oat cultivars. His current research interests include mapping of genes and quantitative trait loci associated with biotic and abiotic stress tolerance, end-use quality characteristics, germplasm diversity and genetic distance, yield per se and synthetic wheat. Dr. Ibrahim has published over 100 refereed journal articles. He also contributed to the procurement of almost $40 million in competitive funding. And he is the fellow of the American Society of Agronomy, Crop Science Society of America, and Texas A&M AgriLife. He is also the chair of the advisory committee for Texas A&M AgriLife Agri-Genomics Laboratory. Please join me in welcoming Dr. Ibrahim.
Thank you. It's a great pleasure to be here. Thank you to Dr. Salh, who made it possible for me to be here. So I attribute a lot of my success to him. So I did my MS here between 1992 and 1994. Then I went to Summit in Mexico, which is like a lot of people say this is the Mecca of wheat breeding. So I stayed for like 1 year and a half, and then I went to Colorado State. And I obtained my PhD and the GMQuick in wheat breeding and genetics. And then I went for my first job as an assistant professor at South Dakota State University. And then I went to Texas A&M, 12 years ago, professor and then a Regents professor. And I'm going to talk a little bit about some of the current trends in genomics and phenomics that we have in our program. And again, what I do is attributed to a team. You cannot do anything by yourself, especially if you are in the US. So a lot of collaboration. So this is my idea about a successful wheat breeding program. What do you need to have? You need to have the diversity in terms of climates. And in Texas, we do testing in about 30 locations where we have yield trials. You need to have stresses, and we have abundant of that. You need to have diverse germplasm, breeding methods, and a diverse team. In terms of innovation, you also need to have germplasm, because we breed for bread quality, but also for flat bread and tortilla type breads. And then also you need to have some innovation in terms of how to bring in genetic diversity into the germplasm. We use gene editing, and I'm going to give you an example of that. And then, if you want to be successful at breeding, you need to have high throughput. And so we have high throughput in terms of using markers, in terms of using image-based selection, because we fly drones, and that facilitates actually the production of images that we can use in selection. And then, we are also utilizing high throughput quality testing in the program, as you can see here. So Texas is a large state. It is the second largest in the country in terms of size, but also in terms of population. And in terms of precipitation, there is only 6 inches of rain here in this area of El Paso, about 20 inches of rain in the high plains of Texas, and then rain increases as you move east. This is where I'm based, College Station, so we have about 45 to 50 inches of rain. So a lot of diversity in terms of rainfall, a lot of diversity in terms of temperature. You have semi-tropical climate here and temperate climate in the high plains. So a lot of diversity in climates, and a lot of diversity in terms of soils. These are the black soils, the cracking vertisols in the Dallas area. You have acidic soils here where the pine trees are grown. You have saline soils along the Gulf. And you have some of the soils that suit wheat production in the high plains of Texas and rolling plains of Texas. So we believe that one of the reasons behind the success of the program is a diversity of climates and soils in the state of Texas. And as I pointed out, we do testing in about 30 locations. No program in the US can test under 30 locations in one state. We do that because we have the infrastructure and the funding to do it. Statewide goals include yield, of course, and then quality is really of paramount importance. We put a lot of emphasis on quality. Some of the wheat grown in Texas is dual purpose, so they can graze it in the fall, and then take out the animal, let it elongate and produce grain. So we focus also on forage production. And then we have a lot of disease, especially rust in South Texas, and we have some pest problems across the state. I put a lot of effort into breeding for resistance to rust. As I said, we are right at the forefront of Puccinia. Puccinia is a genus for rust. So the inoculum survives the winter in the south, and then it gets blown in the spring by wind to other wheat producing areas in the country. So my biggest focus is rust, because I try to develop resistant cultivars that can be grown in Texas, reduce the inoculum that can be blown to other areas. And we have what we call the multi-state leaf rust nursery in the San Antonio area. A lot of my colleagues from different colleges in the country, the USDA, and the private sector, they send their germplasm, I grow it for them, and they come and rate it in April. So what we do in Texas ends up actually impacting the whole country. Well, a picture is worth 1,000 words, as you can see here. I don't know if the light is affecting the quality of the picture or not, but I hope you can see it. But this is a variety trial. These are 5-foot by 15-foot plots. Greenbug has eaten everything, okay, except for those cultivars that have one gene, GB3. One gene makes a difference between a plot that is existent and one that is just gone, consumed by the bug. So Texas is a center of excellence for green bug research. We have cloned the gene. We have perfect markers to use it for. This is also a picture in the Blacklands of Texas. And what you see here is a pressure of Haitian fly. There are about 33 genes of resistance to Haitian fly. Only 4 work in Texas. So this is gone, or a lot of the plots gone. Only those plots that have genotypes that have either H13, H21, 25, or 26 survive in Texas. So we put a lot of emphasis into resistance to disease, but also to insects. And as I said, this chromosome, this gene GB3 lies on the 7DL, so it comes from Aegialopstochii. The story is, there is a long story about that. That gene came from a synthetic wheat that was developed in North Dakota. That one year, there was a huge green bug pressure, and all the field in North Dakota was taken. I mean, it was consumed. There was only one standing row, and they harvested it, and it became cultivar Largo, spring wheat. And so that became a source for green bug resistance that we have utilized heavily. But the bottom line is we have a perfect marker, flanking markers actually of the gene that we use in the program. Heat stress is also a problem in South Texas, and I have trained with my colleagues, Dirk Hayes, too many students. That was a publication producing machine actually, heat stress for us. But also it's a big problem in the state of Texas. This is a breeding pipeline. We use what is called the modified bulk method. We have 4 generations of bulks, and then we derive heads at the F4. We grow 90,000 head rows, and I don't think there is any program in the country that can grow 90,000 head rows. It just sounds crazy, but breeding is a numbers game. The harder you work, the more you test, the luckier you get, and so it's like a funnel. Every time the number of lines decrease, but the number of locations where we test increase, the number of replications increase, and hopefully at the end of this pipeline, we hope to release 1 cultivar. This is a continuous thing. Every year we release a new cultivar to the producers, not only in Texas, but also in other areas in the Great Plains like Oklahoma, Kansas, Colorado, and Nebraska. Well, this is all data, but I just want to show you that a cultivar developed in Texas, TAM 111, at one point had 3 million acres in the country. It was a number 1 in Texas. At some point, it was a number 2 in Kansas, number 3 in Colorado, and number 2 in Nebraska. So oftentimes, what we develop in the South end up being adapted to in the central and southern plains of the U.S. and not vice versa. You know, if you bring a cultivar from Nebraska, it will not do well in Texas. But for some reason, cultivars developed in the South end up doing well in the South and in the central part of the country. So in terms of impact, we have one of the largest wheat breeding programs in the country, as you can see. So in my opinion, this is the best phenotyping machine, the combine harvester, right? But you cannot grow too many lines and just rely completely on yield per se. That's why we try to develop tools associated with traits that can lead you to yield per se. And part of my talk is going to be based on that. Another aspect that we have included in the program, we became a center of excellence for synthetic wheat because you need to bring in diversity to the program. You know, cultivar development relies heavily on diversity, and if you don't have it, you don't have any success. So what do I mean by synthetic wheat? Basically, in nature, that happened about 10,000 years ago near the border between Turkey and Syria. A cross happened between Triticum and Turgidum, just in the wilderness, and a diploid called Agelopstochii that they resulted in Triticum aestivum. But actually, this cross was remade in the lab at Simit by Mujib Kazi and Sanjaya Rajaram. And when they crossed some of the diploids and the tetraploids, they produced what they called synthetic wheat. So it ended up actually having much bigger seed size and weight, but it did not have good tillering ability and it lacked the quality. So we have been crossing our conventional wheat to synthetic wheat to bring in resistance to disease, tolerance to biotic stresses such as drought and heat, and also to increase yield potential. And we have made a lot of progress in that regard. By the way, quality is also a very big issue with synthetic wheat. So we have been working very hard to build better quality into synthetic wheats. This is Mujib Kazi, if you know him. So by using synthetic wheat in the program, we were able to increase yield by 20% and forage by 30%. And as I said, the seed weight is highly heritable. So when you make a cross between synthetic wheat and conventional wheat, you need to, in the offspring, you need to select for tillering types, good tillering types, because seed weight has been highly heritable for us. That's what we have been doing. Series of back crosses to synthetic wheat, and then selecting for tillering ability and quality. And we have some cultivars that are about to be released based on that effort. Hybrid wheat, Texas A&M and the University of Nebraska are the only two public programs in the nation working on hybrid wheat. We got some funding from IWeb NIFA to do so. I also got some funding from Texas A&M to do so, to start the programs. So currently we are developing the tools for it, because wheat is self-pollinated and you are trying to force it to behave like cross-pollinating crop. So you need to have females that are highly receptive, that open their stigmas, expose them to the incoming pollen grains. You also need to have good male types that have very good anther extrusion, and also pollen grains that travel far and remain viable to fertilize the females. Okay, this is what I mean by gating. So the glooms need to separate like that, so they can expose the feathery stigma, as you can see here. And in my program, I have evaluated almost the whole germplasm for the floral traits, because you cannot only rely on hybrid vigor, you have to have the best parents and then look for hybrid vigor later. So these are the traits we look for good anther extrusion and pollen dispersal, good floral morphology in terms of females. They need to have good opening. The stigma needs to come out and poke out so it can actually receive the pollen grain. You also need to have very good heterosis, meaning that, hybrid vigor, meaning that the offspring need to be better than both parents. And so parent development is a continuous effort. So this is some of the data we have in the program. This shows you the highest heterosis as compared to the mid-parent, which is the average of the two parents, or the better parent, or even to the best, to the cultivars that are broadly grown in Texas. And if you look at the whole pool, the highest heterosis we got was 118. Basically, you need 15% heterosis for this technology to work, because production of seed is a bottleneck. It costs a lot of money to do so. So to offset that, you have to have good heterosis. You need 15% minimum. So we have parents that have as high as 118% heterosis when you compare them to the high parent, mid-parent, or almost 100% when you compare them to the high parent, and about 44% compared to the best check. And these are the means across the locations where we tested hybrid in the state of Texas. So we can have 15% heterosis in the germplasm. So, while quality is of paramount importance in our program, we are a quality conscious program. Look at these 2 loaves of bread. They came from 14.5% wheat, but I would rather eat this one, right, and sell it, because it is not only protein quantity, it is also protein quality per se. And we do a lot of testing for that. We also, we also focus on tortilla. And the same cultivar that you develop for bread types will not work for tortilla. For the shelf life, you need high protein, right? Because people buy the tortilla and they put it in the fridge for like 2 weeks before they remember to take it out and put some ground meat and eat it. When they do so, if it doesn't have the good shelf life, it will crumble, right? So we need the high protein for the rollability and the shelf life. But the problem is the high protein will make it retract. So those tortillas that have high protein end up being very small. The manufacturer doesn't appreciate that. So they wanted us to come up with wheat that can develop good shelf life, but also big enough, because that's the bottom line. They're not going to sell very small tortilla. What we did at Texas A&M, we knocked some of the gliadin and the glutenin subunits, and we have very good shelf life and very good diameter. And this is a patent pending technology that only Texas A&M has. We are getting ready to release the Ferris tortilla line very soon. So what I'm trying to say is, we do a lot of testing for quality because we are a quality conscious program. As I said, we have a medium throughput lab for, basically this is just for marker assisted selection. We have a number of Cas-based markers to complement what we do in the field and to increase gain from selection and to increase the breeding efficiency. Okay, but when we want to map a new trait and develop new markers, we use sequencing. We use what is called RAD-seq technology. It is more, using much more expensive machine like that from Illumina. That's an Illumina HiSeq 2500. Okay, and so once we find a good sequence, we develop a marker around it, and then we can use it in this medium throughput lab. Actually, this was one of my students. He is now a professor. So we have somebody else in the lab. Genotyping, we do a lot of genotyping, and again, this is support research. So we don't do research just for the sake of research. We do it to support something in the program. So we need markers, and so we continuously develop new markers. We do a lot of that work through students. So students come and pick a trait of their interest, and they have populations. So they phenotype them, and then we genotype them, and at the end of the day, they put the phenotypic data and genotypic data together to map a new trait. We have about 33 markers for high-value traits that we routinely use in the program, and we have to continuously look for new markers as we go. Now, this is a topic that is really hot. We have become a center of excellence for UAV work. Basically, we fly drones. We can fly a field of 30 acres in about half an hour, and the next day the data is available at the fingertip of the breeder. So we call it unmanned aircraft system, or UAS. We also have some ground-based systems. Basically, you take many terabytes of data in the visible range and in the NIR range. And as you can see, these are different cultivars. You can see the variability in the visible red, green, blue area. You can also see variability in the near infrared. Leaf rust, for instance, destroys the chlorophyll, okay? And that destruction can be captured using UAVs. So basically, some of the goals in the program is to use the UAVs for disease phenotyping, for yield estimation, as well as evaluating drought and heat. And also, all of this is based on what happens to the leaf in terms of destruction that can be captured by those images. So I coined the term image-based technology. A lot of people just call it a high-throughput phenotyping, but I like to think of it as an image-based selection. So what do we do? We have different systems. So you can fly this type of a drone, or you can fly this type of a drone. Here we have a slant range camera, and here we have an RGB camera. The slant range can give you something like NDVI. The RGB can give you anything in the red, green, blue area. You have to have calibration panels on the soil. You have to have those control points in the soil, okay? And the beauty of it is I don't have to do this myself, because we got a big grant at Texas A&M to join the engineers, the computer scientists, and Dr. Muhtar knows about that when he was there. So the engineers, I never worked with engineers before, but through this project I became friends with them, computer scientists. So they work with level 0 data, and what I get is level 2. So what I get is just digital number, right? A number for height, a number for disease that I can easily see, including in my database. All of that work in the background is done by somebody else. We train too many students on this. So we have done some work on the variety trials, and I'll show you here what we have done. So we have made great progress in terms of disease screening. For me to go through a field of 30 acres of land and try to rate like 20,000, 30,000 lines for rust, it will take me 2 weeks or 3 weeks. But we can actually capture that with the drone in about half an hour, and then the next day the data is available at my fingertip. So what we do, this is what we usually do when we take visual measurements. We take notes on the infection type. As you can see here, resistant, that means it has necrotic areas, which is hypersensitive reaction. Basically, the plant tries to kill part of its tissue so it can get rid of the fungus. And then as opposed to something like this, you have a lot of uridinous spores, okay? So the plant cannot, this is highly susceptible, as you can see, or you have a moderate resistant or moderate susceptible. So what we do, and these are the severity scores from 0 to 100%. So I took notes visually on the severity and the infection type, and then I multiplied them by each other. I developed what is called the coefficient of infection, okay? And then I took the average across many readings, and we call this the area under the disease progress curve. So this is my visual data, and then we have the drone data, okay? Of course, you have to go through a series of software to get the data out and give level 2 data to a person like myself. Of course, I don't have time to do all of that. That is done by the engineers and by the students whom we train. So these are some of the equations, like the excess green, like the red, green and blue. And also I know that you know about the NDVI, which is the normalized difference vegetation index. Basically, this is near infrared minus red divided by the sum of near infrared plus red. The higher the NDVI, the healthier the canopy, okay? You get low NDVI readings, and that's an indication of poor canopy. It can be for disease or survival of the winter or something else. So these are some of the equations. As one of the field, very close to Temple, Texas, this is McGregor, Texas. That's about 40 acre of land, so very close to 18 hectares. So when I put together the data, this is my data, my visual data, the data that I took, and that is what the drone took, okay, the RGB camera took. And this was done on the uniform variety trial that has 40 entries. Look at the R square, very high. If you take the correlation of that, it's going to be like 0.92 or 0.93. That means I can actually use this technology to call out what I don't need, to call out the poor types, and actually. So what does it mean? Let me go to the next slide. Here on the x-axis, that's my visual data. On the y-axis, the NDVI reading from the slant range camera. And we have an R square of 0.86, which is a correlation of 0.91, which is a good correlation. So what does this mean to me? It means that actually we can now, instead of 90,000 head rows, maybe you can like double that amount of lines, because breeding is a numbers game. The harder you work, the more you test, the luckier you are going to get. So I can use it to call out maybe 20%, 25% of what I don't need, even 50% of what I don't need. That is, an increased gain from selection and selection efficiency. Okay. Now we're not there yet. You think that we like to use this for Hyatt and stand and canopy cover and all of that, but we believe actually one day we can use this for yield. As I said, we have 90,000 head rows at the F5 generation. It is impossible for us to harvest every single row, process a seed, and then take a measurement on yield. That's why we use art, because breeding is both art and science. Art comes into play here at the head row, and we use visual selection. But we believe that if we use a drone, okay, with one of those cameras, slant range or RGB camera, we can actually count the number of tillers. It can count the number of tillers per plant or per row. It can give you the head size, the length. We have an engineer by the name of Alex Thomason who actually used elliptical shape algorithm, the length and the width of the head, and he came with an R square, a coefficient of determination of 0.7 between yield and the dimensions of the head. So one day maybe we can actually use this to select for yield. Okay, we're not there yet, but the future looks very bright. Now I said that we need to increase diversity. We use markers, but we can actually use precision breeding by modifying specific sequences in the genome. We can bring in a new sequence, or we can knock out function that we don't need. And that CRISPR-Cas technology was taken from bacteria, because this is just a bacterial cell. Bacteria is usually attacked by specific viruses called phages. Basically, they eat the bacteria. They come inside, they penetrate the cell, and then they steal the machinery of the bacteria, replicate themselves, and then they start literally eating the bacteria. So the bacteria developed this technology a long time ago by recognizing the genetic sequence of the invading phage. Okay, it becomes part of the genome of the bacteria. So once it becomes part, it can transcribe RNA for it, right? Depending on what phage there is. So different transcriptions. We call these guide RNAs. So when the phage comes, the guide RNA with the Cas9 complex, Cas9 is nothing but a protein that has a nuclease activity, meaning it has scissors that can cut the virus. So it finds the virus by complementarity, the sequence pairing happens, and then the Cas9 protein cuts the virus and kills it. Okay, so it becomes part of those repeat sequences, spacer sequences. They become part of the phage of the bacterial DNA sequence. So when a new phage comes in the future, it is easily killed by the bacteria. So the technology has been taken from that bacteria. Scientists at US Berkeley and Harvard Institute found that we can actually use this technology to bring in new genes or maybe knockout functions. And so I'm going to give you an example from my program. Oops, sorry. Okay. So as I said, successfully used in Arabidopsis, maize, rice, sorghum, and wheat. We don't use agrobacterium. We use actually the particle guns. So we call it ballistic delivery system, which use gold particles. So you attach the DNA sequence, and then you insert it into the genome of the wheat. We tried to infect protoplast tissue. It didn't work because protoplast has only a couple of cells. So we ended up actually transforming immature embryos, and I'll show you that in a minute. By the way, this is TAM 114. This is the best hard red winter wheat in the country, developed in our program, statewide program. So this is a field, this is a farmer field, and this is a variety trial, and we have taken a picture by the drone. So TAM 114 is a high yielding line, has good resistance to disease, and one of the best qualities of any hard red winter wheat, but it is red. And so we wanted to develop a white version of it. The same endosperm, but we wanted a white brand because white wheat has a premium. You can sneak the fiber that way, make it a healthy product. It can sell for a premium that way. You can get better flour extraction. So how do we do that? Realizing the fact that the red color gene is inherited in the 3 genomes, the A, the B, and D genome. So to have a white version, you need to knock down the 3 genes, because one gene red will give you red brand. So our task was to knock out all of those three genes in the A, B, and D genome. Okay, and so we developed guide RNAs, and the idea is to knock down the function. So it's, by the way, in gene editing, it is easier to knock down a function than to bring in a new trait. Okay. So this is part of the anthocyanin proanthocyanidine pathway. So these are, the bolded symbols actually refer to enzymes. And there is a gene called TAMB110 that upregulates all of those genes in the pathway. So you have to go through all of this pathway to produce a red pigmentation. So if you actually break this pathway anywhere in the cycle, you knock down the red gene function. So that's what we did actually. Okay, so red grain code is composed of pigments like this and controlled by the gene TAMI B110 located on the 3A, 3B, and the 3D genome. Remember that wheat actually has the ABD genome. A and B came from Triticum turgidum, which is a pasta wheat, if you call it Triticum durum, which is a relative of Triticum turgidum, and the D genome came from Aegilox toshii, which is a grass. So red color is controlled by 3 genes located on the 3A, the 3B, and the 3D. Okay, so that's our task, is to try to interrupt this pathway. That's what we did. So in the first picture, you see 3 exons of the homologous genes amplified, and this is an electrophoresis product, and this shows the amplification and the sequencing. So we separated those genes, and then we sequenced them. There are 3 copies on the A genome, 3 copies on the B genome, and then we can only do 2 copies on the D genome. I'm not showing the exact sequence because it's at this point we cannot show it until Texas A&M permit us to do that. You know how things go there in Texas. So remember that we have 3 exons, and you know the difference between an intron and an exon. An exon is part of the chromosome that codes for gene. So we wanted to look for a conserved sequence for every one of the genomes, the 3 genomes. You don't need to do the 3 exons. If you can knock out one of the exons, that's it. And so that's exactly what we did. We sequenced those areas, and we found them to be very conserved. And now our task is to develop the guide RNA and knock them out and produce a wide bran wheat. So we did that. We transformed immature embryos. You have to excise them and do that under the microscope. And then we use the gene gun to insert the guide RNA into the sequence, and we did that on 266 immature embryos. I'm not going to go through all of those details, but ultimately you have to take them into an osmotic medium like that, and then a recovery medium, and then you have to, this is another recovery medium, and ultimately a regeneration medium, and then you put them in some rooting medium. It takes a lot of work until you take them to the greenhouse like this. So the plant I'm going to harvest them when I go back actually to Texas A&M University. He's a student who worked on this project. His name is Jorge. He's from Mexico. He came us through the CIMIT program. Rabi Singh is a friend of mine, so he sends me really good students to do the work. So hopefully when I go back and harvest the wild grains, I can get back to you and tell you that we have done it, okay? So basically this is a glimpse of what we do, and it takes a team. We meet every year. These are all of the professors. The leaders of the team are myself. I don't know where I am here. That's me, and Jackie Rudd somewhere, and then we have a number of professors and postdocs and students and administrators. So 1 + 1 is not equal to 2. If you work together, it is 3. And if you antagonize each other, 1 + 1 is a -2. Okay? So we do it by being a team. And that's what I have to tell you today. Thank you so much.